So my name is Crystalyn Partica, and I'm the project manager for the Northern Grapes Project. One of my responsibilities is to host the webinar series. Today's webinar is Selecting Yeast for Cold Hardy Cultivars, presented by Katie Cook of the University of Minnesota. Katie is the Enology Project Leader, and her appointment is split between research and outreach activities in the state. Her main research is focused on enological evaluation of potential new grape cultivars developed through the University of Minnesota's grape breeding program. Chris Gerling is on the webinar tonight. He's an extension enologist at Cornell, and he's going to help us with the questions in the chat pod. Remember that as a participant in the webinar, you will not have the ability to speak. If you're having any technical difficulties, please type a note in the chat pod over on the right side of your screen, and I'll do what I can to help. If you have questions, please type those in the chat pod as well. Chris will handle some of them as the webinar progresses, and we'll save some of them to the end for Katie to address. So with that, I am going to turn things over to Katie and let her get started. Katie?
Okay. All right. Can everyone hear me okay? I think there's still Jim having some audio problems, but other than that, I hope everyone else can hear. And thanks everyone for spending your evening with me during prime Christmas shopping hours on a Tuesday night. We decided to do this yeast selection webinar series just because it's one of the main questions that we get asked from winemakers, and the main purpose of this webinar will just be to try and alleviate some of that confusion that people seem to have and some of the anxiety that they have over choosing yeast. So, as always, before we talk about the present, it's important to understand a little bit of the history of yeast. So we know that yeast from the Saccharomyces species have been used for thousands of years for fermentation of food and beverage, and this is just from genetic evidence and along with some physical evidence that exists about yeast. However, we don't really know when people started purposefully adding yeast to their fermentations, whether it be wine or beer or bread, because it wasn't really until 1860 that yeast was discovered as being the organism responsible for changing sugar into ethanol and responsible for fermentation. And it took even another 30 years before Müller-Thurgau started recommending that we should inoculate wine with pure yeast strains to ensure proper fermentation. Most of you probably already know this, but wine is also a very old beverage from at least 5000 BC, and sorry, I'm trying to get my pointer out here. So, and one of the reasons we know this, because archaeological evidence, they found these old amphora in around Mesopotamia, I guess, but it has these crystals in it that are potassium bitartrate crystals. So it's the same crystals that you see forming in wine today, and as grapes are one of the few species that actually forms, or actually has tartaric acid, we know that this container must have held wine at some point, because the potassium bitartrate is only formed when there's that supersaturation. When people used to ferment wine, they used to fill their vessels up and ferment the wine in the vessels, and then after fermentation was finished, it was actually sealed off and marked with the date, name of the vineyard, a person in charge of the wine. And they would really only crack it open over special occasions, because wine, the process of fermentation in wine happened so reliably, and wine basically made itself, so they assumed that it must be something that, like, a gift from God, and so they tried to only open it during religious ceremonies, and they tended to drink beer on a more day-to-day basis. So we know now that the main yeast involved in fermentation is Saccharomyces species, which literally means a sugar fungus, which highlights one of the most important characters of the yeast, is that in the absence of oxygen, it's able to transform sugar into ethanol and carbon dioxide. And we also, through genetic evidence, we know that it evolved about the same time as fruits evolved during the Cretaceous period. And it had this ability to produce large amounts of ethanol, and then they can also tolerate it. And it's able to grow in both aerobic and anaerobic conditions. So a little bit more about the different types of Saccharomyces yeast. So there's a few different species that are found in natural environments, so on oak trees or different wood, and they're not really associated with human activity, but they're part of this sensu stricto species complex. And then we also they've also isolated a species, Saccharomyces uverum, which has been isolated in wine and cider fermentations, as well as Saccharomyces bionus, which is used mainly in lager beer fermentation. And then Saccharomyces cerevisiae, which is the most important yeast for fermentation. It's used in wine fermentation, beer, sake, and then palm fruit in Africa. And it's also the yeast that's used in leavened bread. With all these different species of Saccharomyces, there have been both natural hybridizations of between the different species, as well as variations in ploidy, these hybridizations have occurred both naturally and on purpose by human activity. So a lot of the cultivated yeast are actually hybridizations between some of these different yeast species. So the main yeast, Saccharomyces cerevisiae, a little bit about this. So just some fun little information about it. So there's been a number of different strains that have been discovered throughout the years that have been isolated from different beverages and food products. But there really haven't been very many different strains that have been found in nature. And so there was confusion as to whether, which came first, the chicken or the egg. So did it first appear in nature, or did it first appear throughout human activity and then a select few went through nature? And we know now that we're pretty certain that the origination was in natural environments, and then this natural origin of Saccharomyces cerevisiae was then followed by human domestication. And if we look at just strains of Saccharomyces cerevisiae that were isolated from wine yeast, we can see that 95% of the strains in the world belong to the same genetic cluster. So the importance of this is that it really suggests that there was a unique origin of wine yeast followed by expansion of populations. So right now it looks like the initial domestication may have been located in Mesopotamia, and then there were two migration events that happened, and the routes can be drawn through Europe. So first, an area close to Lebanon, and which migration occurred through the Mediterranean Sea toward Italy, and then through Spain and France. And then in France, there was another migration route through the Rhone Valley toward Burgundy. And this sort of transfer of yeast strains may have been related at least partly to movement of different grape varieties as humans migrated and transplanted grapes. And for example, one of these grape varieties, which is the most planted in France and Italy. In Italy it's known as Trebbiano, and in France it's known as Uni Blanc, which most people are probably not very familiar with it. But this grape started in Italy, and it's one of the authorized varieties in Châteauneuf-du-Pape as well as being an important variety in Cognac and Armagnac production, and the most important the most important grape in the Loire Valley for Muscadet production. All right, so to end a little bit of the history, so now just to start talking about what happens when you ferment a wine with yeast. And this is the most important when we're talking about about commercial yeast or inoculating wine with yeast is what exactly does the yeast contribute to my wine? And we know very well that yeast contributes ethanol, so it's part of that fermentation, transforming sugar into alcohol. So yeast are also direct producers of glycerol, which can add some mouthfeel and body to a wine that's lacking in it. And different strains produce different quantities of glycerol under certain conditions. They also produce a class of aromatic compounds called higher alcohols. Now, there's only really one higher alcohol, phenylethanol, that has a positive aroma that smells like rose. But for the most part, higher alcohols smell pretty bad. Most are described as having a solvent-like or hot aroma. Methanol smells a little bit like cabbage. And most of the time these higher alcohols, they're also known as fusel alcohols, which tells you a little bit about some of what they might smell like. But most of the time they're present in pretty low concentrations in wine, so their impact is fairly minimal. In higher concentrations, close to 400 parts per million, they can start masking other aromas in your wine. If we want to look at some of the winemaking parameters that increase the production of higher alcohols, we know that high pH, elevated fermentation temperatures, so warm ferments, oxygen exposure during winemaking, are all going to help contribute to this higher alcohol production. In red winemaking, sometimes it's difficult to limit some of these factors due to pumping over and some of the elevated pH issues. But it's just something to be aware of. And in white winemaking, if you ferment at sort of ambient temperatures or the warmer temperatures, kind of like 68 to 72 degrees, that will also limit some of the higher alcohol production. Another important aromatic compound or a class of compounds are esters, and esters have really fruity aromas like banana and strawberry. So if any of you are familiar with Beaujolais Nouveau wines, they're usually pretty high in esters, and these are also used as flavoring in the candy industry and in a lot of other foods. So those are a pretty important class of compounds. And yeast also produce really small amounts of acetic acid and lactic acid. Acetic acid production might be from 100 to 300 parts per million. Lactic acid might be 200 to 300 parts per million. The important thing to remember with acetic acid production is that it will increase with your initial sugar concentration. So high sugar musts are always a little bit higher in acetic acid than musts that are lower in sugar. So, and this is really independent of the quantity of sugar fermented. It only matters about the initial sugar concentration. So this is part of the reason why late harvest wines tend to have a higher VA. Other factors that can increase the acetic acid production by yeast are rot, so like if you have botrytis on your fruit, very low or very high pHs, so less than 3.1 or above 4.0. If you have certain amino acid or vitamin deficiencies, high fermentation temperatures can also increase acetic acid. And this is especially true during the initial yeast multiplication phase, so right after you inoculate your wine with yeast. You need to make sure that your temperature is cold at that inoculation phase, or cool, so you probably shouldn't exceed 68 degrees at the start of alcoholic fermentation, if you can help it. And there's a lot of indirect aroma contributions by yeast as well, which are probably some of the more important contributions. One of these is that they enhance varietal aroma by freeing some of the bound aroma compounds in grapes. And one of these class of aroma compounds are called monoterpenes, and these are very floral aromas. They'll smell like rose or lilac, and they're really in high abundance in muscat varietals or other aromatic varietals. And you can actually, if you eat muscat, you can taste these aromatic compounds in the grape, and it's what makes muscat type grapes taste so yummy and have this floral aroma. So we call those that you can taste, or maybe you smell it in the juice during processing. We call those the free aroma compounds because they're free and they're free in the juice, and you're able to smell them. But there also is a certain quantity of monoterpenes that are bound to essentially sugar molecules, and yeast help cleave they enzymatically cleave these. I did this in the first one. They enzymatically cleave the bound compounds or the aroma precursors. So they allow these bound aromas to be free in the wine, and so So during fermentation, you'll actually increase the floral aroma of your grapes, or of your wine. I'm sorry. Thiols are another class of compounds that also exist in wines, and we know, and they're especially important, and this has only relatively recently been discovered for Sauvignon Blanc, probably in the last 10 years. The thiols are very important contributors to the sort of tropical fruit aromas like passion fruit and guava, or maybe grapefruit aromas, and then sort of a lemongrass or box tree aroma that's associated with Sauvignon Blanc wines. So, and yeast have the ability, well, let me backtrack a little bit. So thiols rarely exist in a free state in the juice. So if you're eating a grape, if you're eating a Sauvignon Blanc grape, it doesn't taste much like what the wine will taste like, because it's over the course of fermentation that some of these aromas are released into your wine and contribute to the wine. So for Sauvignon Blanc, yeast selection is actually fairly important for the wine aroma, and again, this has only been a recent discovery even for Sauvignon Blanc that's been around a long time. So we're starting to learn more and more discoveries are made every day about these aromatic compounds and how yeast contribute to them. Not all the contributions are good. Like hydrogen sulfide has kind of a sewage or rotten egg aroma, and yeast can increase your hydrogen sulfide content in your wines. And then spicy characteristics. We know just from sensory analysis that comparing juice to wines, that certain yeast, that the wines tend to be spicier than the juice. There's still some studies determining what mechanisms are involved with that with the yeast. I know there's some studies in New Zealand with rotundone, which is a black pepper aroma, trying to find a mechanism how yeast enhance it. And then there's also others. Like I said, this is it's not really a new study, but I think just with new technology, we're able to look at some of this stuff more closely, and we're learning new things every day about how yeast contribute to enhance the aromas of wines. And then we can't forget about yeast leaves too, because yeast leaves also play a significant role in contributing to the overall aroma of your wine. They can remove some undesirable compounds, so the ochratoxin A, which is the carcinogen, and legally they're limiting amounts that you can have in your wine now. Diacetyl, which has a buttery aroma. A lot of times people don't want that buttery aroma in their wine. When it's produced, it's usually produced by lactic acid bacteria. There's a little bit produced by yeast as well, but it's usually insignificant. So you can remove some of that buttery aroma and clean up your wine. And also fungicides, so residual fungicides that are in the juice, can also be removed by yeast, or by yeast leaves, sorry. They also can increase undesired compounds, so there's some good and bad for the yeast leaves. Some of the undesired compounds are biogenic amines, which You probably know histamine. You're probably all familiar with histamine, which is a biogenic amine, which causes heart palpitations in some people and increase in blood pressure when they drink wine. But other biogenic amines are putrescine and cadaverine, which, if you can imagine, don't have very nice aromas, with names like that. Fatty acids can inhibit your malolactic fermentation. And then the higher alcohols we've already talked about. So those are your fusel alcohols that are going to have those solvent aromas, mostly have solvent aromas. There's some desirable compounds that they can increase as well. So manoproteins are the main desirable compounds. So they add some body and structure to your wine, mouthfeel. And it's also been shown that short contact with a large quantity of lees can increase your esters. However, the lees can also remove some of these esters over a long period of time. So during extended oak aging on lees, you'll remove some of the esters. You'll remove diacetyl, which some people don't like, and you can also remove some of the oak aroma, which, depending on the wine style you're shooting for, might be desirable or undesirable. So all these things the last 3 slides that we've gone over, they're these indirect and direct contributions by yeast lees. You need to think about all this when choosing a yeast strain. So I know I probably haven't helped narrow down or make it easier to choose yeast strains, but this is why it's so difficult to make yeast recommendations, because there's a lot of things that a lot of different things that are produced by yeast. And this is whether or not, you need to think about this whether you're inoculating or whether you choose not to inoculate your wines and try and do natural fermentation. And talk a little bit more about spontaneous fermentations, but for that we'll just to summarize some of the advantages of using cultivated yeast or the active dry yeast. So you get a faster start to fermentation, so you exclude some of the defects due to a delayed start. You get a greater yield of ethanol, so the cultivated yeast are usually more efficient at converting sugar to ethanol. You get a lower production of volatile acidity and other off aromas. You get full exhaustion of fermentable sugars, so usually you don't have these stuck fermentations where there's some residual sugar at the end that then can be used as a carbohydrate for bacteria growth and lead to other instability issues. You also can, now we know some of the other attributes of yeast, of cultivated yeast, so some of them are able to consume malic acid, so you might get some better control of your total acidity by partially degrading some of that malic acid during fermentation. And we also know that through cultivated yeast we can determine which strains optimally produce higher alcohols or esters or maybe glycerol, so if you want to enhance certain attributes of these secondary metabolites. And yeast, you're also going to optimize your interaction with the malolactic bacteria. So there are certain yeast strains that are more compatible with various lactic acid bacteria strains. Some of them are incompatible together. And by knowing this compatibility, you're going to have less issues with problems with malolactic fermentation if you decided to do that later on. And also, since we've started using cultivated yeast strains, it's actually made wine safer. You can, because they reliably start fermentation at the beginning, we're able to start out with lower levels of SO2 from the beginning. Some people use, as low as 20 parts, 10, 20 parts of SO2 at the beginning of fermentation. Yeast, especially the yeast lees, can remove certain heavy metals and some of the carcinogens that we talked about before. And also the cultivated yeast, we know that they're lower producers of ethyl carbamate, which is a carcinogen as well, and the biogenic amines like histamine. You also are going to have a regular fermentation pattern, so when you inoculate with the cultivated yeast strains, you'll have regular growth phases of yeast. So usually you have a really short leg phase in the beginning, and during this leg phase, this is where your yeast populate. You'll have aerobic respiration. Usually there's quite a bit of dissolved oxygen because you've just crushed all your grapes. And so the yeast will go through aerobic respiration, and this is sort of they start multiplying and increasing their population. As the wine becomes more anaerobic, this is when you start to see fermentation really kick off, and the yeast are growing. They're doing a little bit of aerobic and anaerobic growth. And then you'll see the yeast slow down, deceleration. This is because the yeast population has reached its maximum. Some of the nutrients have been depleted. And then you'll see a stationary phase. And this is, the yeast viability is usually pretty high at this stage, but their activity is pretty low because they've depleted most of the sugars, and the ethanol levels are starting to become toxic. And at this phase, if you're starting to get a stuck fermentation, a lot of times if you just do a quick racking of your wine and add a little bit of oxygen, it can kickstart the yeast a little bit and let them finish off the fermentation. So now a little bit talking about spontaneous fermentations, or these natural fermentations. And the natural fermentations are carried out both by yeast on the grapes and on winery equipment. And however, the yeast population that's present on grapes are usually non-Saccharomyces yeast. So there's many different genera of yeasts that exist in wild fermentations or naturally on the grapes. And there's been a lot of studies looking at these native yeast populations in vineyards and comparing them to the winery. And most of the studies have concluded that that the Saccharomyces cerevisiae is absent or rarely present on grapes. One study I read talked about 1 in 100 grapes having Saccharomyces on it, existing on the grape surface. But the Saccharomyces is usually highly associated with the winery equipment, so in the wine cellar itself, in tanks, on presses, and different equipment. And so usually the origin for Saccharomyces cerevisiae and spontaneous fermentations is in the winery itself. And just a quick look at some of the other species or genre of yeast that can be present in a fermentation, and some of these can contribute positively to your fermentation. So the Hanseníaspera has been known to be a producer of some positive compounds, like it can produce some positive volatile compounds like some esters and ethyl acetate, which the ethyl acetate can actually contribute in low quantities. It also produces glycosidases, which contributes that cleaving of aroma precursors. The Torulaspera contributes a lot of these glycosidases that will cleave the aroma precursors and increase some of the monoterpenes and thiols in your wines. Others aren't so positive. Pichia yeasts are normally, Pichia and Candida are normally some of the yeasts that are associated with the floor yeast on top of your wine. And of course, most of you probably know about Brettanomyces that leaves the volatile phenols, so the horsey aromas, the horsey off aromas in your wine. Anyway, so there's some benefits to some of them, and then some of them can cause some problems with the wine. And the problem is we don't really know, unless you do an analysis of the population of yeast, you don't really know what's existing out in your vineyard. So it's always a little bit risky to do a native yeast fermentation. There are some good things that happen with native yeast fermentations. One is that because there's a higher population of this non-Saccharomyces yeast, they usually get going pretty quickly. So even though there's this lag phase when the Saccharomyces is growing, the non-Saccharomyces will take over and prevent bacteria problems like Acetobacter and other things from taking over your must and causing some off aromas. And these non-Saccharomyces yeasts, they usually will ferment up to about 5% alcohol. Most of them aren't tolerant to alcohol beyond that point, so they'll just naturally die off. And at this point, then it's just sort of a natural selection, and the Saccharomyces yeast will then dominate the fermentation. There's been a lot of studies making this comparison, aroma comparisons between cultivated yeast and native yeast. So this is a study done by Bruce Auckland, and he looked at some of these highly aromatic compounds, like this first box that we're looking at, the monoterpenes, so those floral aromas that we talked about earlier, and he compared various cultivated yeast strains, so the Prise-de-Mousse, D47, Fermi Blanc, VL1. The VL1 and Fermi Blanc are known to be pretty aromatic and increasing these some of these compounds. And then he also used a native yeast fermentation, and you can see that in some cases, with some of the monoterpenes, the native yeast did better, and others it didn't. And if you look at overall the monoterpenes down at the bottom, it's actually not any better than some of the other selected yeast strains. And actually the Fermi Blanc showed a really high quantity of these monoterpenes. He also looked at some of the positive higher alcohols, so the ones I mentioned that have this rose aroma. And with those it was actually fairly high, so the total amount of these aromatic alcohols, or the fusel alcohols, were 140. However, he only looked at 2 of them, and so it's uncertain whether there were some of the negative fusel alcohols that also contributed to the overall aroma of the wine. So again, just looking at those totals, but it's just to point out that there's some advantage of native yeast, but the cultivated yeast also can have these same advantages. So some of the bad things, the main thing is that you're going to have stuck or sluggish fermentations, mostly because your initial Saccharomyces cerevisiae yeast is the population is low. Some of the off aromas, you can have higher production of volatile acidity and some of the higher alcohols that I mentioned. And you can also have malolactic fermentation start spontaneously in the middle, which might be desired, but it might not be desirable as well. And some of these can also cause inhibition of malolactic fermentation later on if that's something you desire. So if we go back to looking at the fermentation kinetics when you inoculate, you have this sort of regular fermentation. When you use native yeast, you can have sort of more irregular patterns. Oftentimes it's a longer lag phase while the initial population is developing. But during that lag phase, you'll see this non-Saccharomyces activity. So it might look like the fermentation is starting off normally, when actually your population of Saccharomyces yeast never reaches the full potential. So you can often get stuck fermentations at that point. However, a lot of the yeast companies are recognizing some of the positive attributes of these non-Saccharomyces yeasts, and we're starting to get more cultivated yeasts that are non-Saccharomyces. So there's at least one company that makes a sequential inoculation with Torulaspora, where you can add that and then add a Saccharomyces yeast a couple days later. The deacidification yeast, so the Saccharomyces pombe, has also been used extensively. And then also with yeast, sort of inactivated yeast products to increase your manoprotein content of your wine, or there's other yeast that might increase glycerol production if you want to increase your mouthfeel. Okay, so now sort of the heart of this this presentation is kind of, so how do you choose a yeast? So we've seen that it's really complex. all these yeast products that are produced during fermentation. And so this is why when people ask me what yeast should I use for Marquette, it's very, very difficult to come up with one answer, because there's actually a few things that you should consider before you decide on what yeast to use. And the first question you should ask yourself is, what style of wine would you like to produce? So you're going to use a very different wine if you want to make young, fruity, fresh wine versus a barrel-aged reserve wine. You're going to be limited by yeast selection with depending on your harvest chemistry as well. So some really high Brix months, or if you have a high potential alcohol, it eliminates some of the weaker ethanol tolerant yeast. And cellar limitations are another thing. So if you don't have cooling systems or some of your analytical capabilities, and then also the post-fermentation treatments. So if you're planning on doing malolactic fermentation, you want to look at yeast compatibility with your lactic acid bacteria. Or maybe if you're doing extended aging on lees, you want to look at different types of yeast for that. And it's always important to remember that yeast is simply a tool, and it's a tool that can help drive a wine style, but it's just a small part. And really, and I know you've probably heard this a lot, but the quality of your grapes is going to have the biggest impact of your final wine, and yeast isn't going to be a cure-all unless the quality is up there. I'm talking a little about, usually when you talk about high-quality grapes, when we're talking about vinifera species, you want to achieve proper phenolic ripeness as well as proper technological ripeness. So some of this, but however, usually in northern climates, we don't often talk about phenolic ripeness in grapes, mostly because we're limited by climate and some of the genetic constraints of our cultivars. So we can't always wait for the perfect phenolic development when there's early frost or a short growing season or just a colder growing season. And then the cold hardy grapes, a lot of them just get really high Brix and high pH early on, so we don't have the luxury of waiting for proper phenolic maturity. And hopefully we'll know a little bit more about this by the end of the Northern Grapes trials. So so again, you're thinking about wine styles. You also need to think the grape variety is important, whether you have an aromatic grape, so like a grape like La Crescent versus a more neutral one, say Prairie Star. The ripeness of your fruit, where it's grown, and then also kind of the age of the vine, or sort of these other quality issues, if the fruit's overcropped or shaded, and kind of your aroma and phenolic development. Most of the time when you're thinking about a wine style, the style is determined more by the quality of the fruit rather than the winemaker. So even though you might want to make a reserve red that's barrel aged for 2 years, sometimes you can't do that when your fruit, or it's impossible to do that when your fruit quality isn't there. So poor quality fruit always needs to be treated a little bit differently. You want a shorter maceration time, cooler fermentation temperatures, and maybe less extractive techniques because you're going to have green herbaceous aromas. And oftentimes you don't have the full aroma development of the grapes, so you need to use yeast that have some high aroma impact to give your wine some good aromas. So let's say you want to make a light and fruity wine. You have some young vines, or maybe you didn't get good ripeness, or you have a high vigor site where you didn't get that good ripeness. So you may want to choose a yeast. Some of the considerations one of the considerations is maybe you're going to lack mouthfeel, so you want a higher glycerol producing yeast. Often you're going to be fermenting these wines at cooler temperatures, so you want to make sure you have a yeast that's going to tolerate the temperatures that you want to ferment them. And like I mentioned before, you're not going to have the aromas in your grapes, so some of the fruity flavors in your wine are going to be derived more from the esters. So at this point, you're going to look for a higher ester producing yeast. And you also want to make sure that the fermentations are finished quickly, and these minimal production of thiols, except the Sauvignon Blanc, because some of the thiols lead to reductive aromas. And let's say you have a little bit higher quality fruit and you're going to barrel ferment, and this is more in the style of barrel fermented Chardonnay. So usually this is when you have a little bit riper Chardonnay, something that has more potential for aromatic complexity. In a barrel fermentation, it's going to be more at ambient temperatures. You don't have to worry about really hot fermentations because a smaller container means that it's usually not going to get carried away from you. And the barrel fermented wines usually spend some time on lees, so they'll gain some body and mouthfeel. So for yeast, because the fruit quality is better, you want a neutral yeast or something that's going to enhance the varietal character. The temperature tolerance isn't as important, and maybe if you're doing it on lees, you want to look for something that has a high manna protein production to increase that mouthfeel. Another one of the main limitations on yeast during a fermentation is the fact that it has the cell wall is made up of a lipid bilayer, so essentially 2 layers of fat. And if you think about cooking oils or fats in your kitchen, you can imagine how temperature changes make those either more fluid or more stiff, and or how different solvents like alcohol might change the fluidity of those fatty acids. And then because these yeasts are passing toxins outside of their cell from the inside out, like ethanol, and trying to import nutrients into the cell, the osmotic pressure against the cytoplasm in their membrane is really can limit their how well they're able to function. So yeast must that has really high sugar concentration provides this great osmotic pressure on the yeast. So you need to take that in consideration, that it's going to be a stress on the yeast at the start of fermentation. And this is more true with late harvest styles, where you want a yeast that's going to have a high osmotic tolerance. And you also need to make sure that your yeast is going to tolerate the potential alcohol of your final wine. So if you have 26 bricks, that's going to be at least 15% alcohol or close to 15% alcohol. So you want to make sure that that your yeast are going to tolerate that. And then also yeast are going to be really sensitive to nutritional factors. So if you don't have the ability to measure the nitrogen levels in your must, you should maybe steer away from yeast that have high nutrient needs. Like I mentioned earlier, some of these dessert wines, they have this really high sugar must, so you need to make sure that yeast have a high osmotolerance or against this osmotic pressure on their cell. And if you look at any catalog that gives yeast recommendations, anything that's recommended for late harvest or ice wines are going to have this tolerance against osmotic pressure. Your alcohol tolerance is going to be that's more of a winemaker style standpoint. So maybe you want a lower alcohol, higher sugar, or maybe you want something with a little bit higher alcohol, and that all depends on sort of the balance you're looking for in your wine. I mentioned earlier that these high sugar musts are always going to have a little bit more VA production by yeast. You want to make sure that the yeast are low VA producers. Again, most yeast that are selected for late harvest wines are low VA producers. And because your fermentation is carried out a little bit longer, you want to make sure that it's not going to produce a lot of hydrogen sulfide. And with fortified wine, it's a little bit counterintuitive because usually you're fortifying up to 18% alcohol, but this doesn't mean you want a yeast that is tolerant to 18% alcohol, because in this case, the idea is that you want to kill off your yeast with an alcohol addition. And if you have a very alcohol tolerant wine, something that is tolerant to 18 might be able to be bumped up to 19% tolerance if the conditions are correct. So you maybe want a weaker fermenter, something that's less tolerant, so you can be sure when you add the alcohol to mute the fermentation that it does in fact kill the yeast. So now going back to the idea that most people ask for yeast recommendations based on cultivar. So what should I use with Marquette, or what should I use with La Crescent? Most yeast catalogs, or the cultivars that are included in yeast catalogs, are actually a really small percentage of the world cultivars, and but they're just some of the most common cultivars, and that's why they're included in the catalog. So most people in the world actually don't have that luxury of looking at a certain grape cultivar and knowing what yeast is going to be recommended for them. You can look at some clues in the yeast catalogs. So there's some keywords that you can look at, like monoterpenes, which we talked about, those floral aromas, or those fruity ester aromas. So that's going to be, if you want a very aromatic, fruity, fresh wine. The thiol producer, usually for Sauvignon Blanc, neutral, so the yeast will have minimum impact on the aromas. All the aromas are going to come from the grapes. Something for aromatic grapes. Extraction is usually for like phenolic extraction, so if you have low color grapes, something that's maybe more extractive. And then anything that increases mouthfeel is probably either a glycerol producer or a manoprotein producer. I'm not going to go over this because I wrote a big article on it that you can look up online, and I'm getting short on time. I'm nearly done. So you can read a little bit about our thought process for our trials that we went over this year for yeast selection. Hopefully by the end of this Northern Grapes project, we're going to be able to give specific yeast recommendations for various cultivars based on some of the large sensory studies that we're going to be doing. Just a few other things to think about are some cellar limitations with your yeast. So I mentioned that if you don't have cooling system in your winery, you need to factor that into your yeast selection. So you probably don't want to get a yeast that's a fast fermenter, because your fermentation might finish really fast and get really hot and blow off all your aromas that you're trying to keep in your wine. If you can't measure nitrogen, so if you have limited ability for lab analysis, you should probably lean toward yeast that have low nutrition requirements. And then, I know this isn't a huge problem in a lot of northern wineries, but if you have large tanks, you see this more in California or bigger industries, there's a bigger reduction potential in those tanks, so you probably want to choose a yeast that has lower hydrogen sulfide production, so you can limit some of those reduction aromas. And now with some of these non-Saccharomyces yeasts or certain strains of yeast that have malic degrading capabilities, so it's a malloethanolic, so they turn malic acid into ethanol. These can provide some interest. We've done some trials with the Schizosaccharomyces pombe on Frontenac Gris, and it has some good results, but it's more the encapsulated yeast product. So you'd insert it in like a tea bag and leave it in for a few days, and then take it out once you achieve the acidification that you want. It's a little bit tricky because sometimes some of the native yeast can take over, and fermentation is going to start before you get the proper malate reduction. And it's also pretty expensive, so it's something you need to look into if that's something you're interested in. And we're also starting to look at some of the Saccharomyces strains that partially degrade malic acid, like 71B is pretty well known, but that's also a high ester yeast, so it can have some wine style constraints. But we're looking at a couple others for some biological deacidification properties. And then there's also been some genetically modified yeasts that have that malate degrading. Another thing that's kind of interesting is the efficiency of fermentation. So We know that wild yeasts are not as efficient at converting sugar to alcohol. Usually, on average, if you have a cultivated yeast, we say 16.8 grams per liter for 1% alcohol. Some of them need a little bit higher, like 18 grams per liter for 1% alcohol. And some of these yeasts are becoming really important, especially with global warming and warmer climates where you get high Brix. And so there's actually a few cultivated strains now that have low fermentation efficiency. So if you have a high potential alcohol problem, it can actually lower your alcohol by 1%, or lower it by a couple points. Anyway, so just some few conclusions. If you understand a little, I hope after this you can understand a little bit more about what yeast do contribute to your wine, as well as some of the limitations of yeast. It's not going to be a cure-all for your wine. And I hope that you're able to start navigating some of these key phrases in the yeast catalogs and guide you toward better decision-making for your winery. And then also feel free to use, now that you know this knowledge, feel free to call the companies that sell yeast, like Scott Labs. They're very knowledgeable, and you can ask some of these technical questions like, tell me what sort of malate degrading yeast you have that would be good for Marquette, and then they can give you more specific recommendations based on your needs. Again, know that yeast aren't going to be a fix-all for your wine, but they can be an important tool for steering your wine in a certain style. It's also really important to plan your yeast selections knowing the limitations of your winery, and to keep that in the back of your mind. And then just finally know that some of the most important aspects for fermentation are going to be your starting Brix, the temperature of your fermentation, and the yeast nutrients in order to have a clean and healthy fermentation. And that's going to have the biggest impact on your wine overall. Okay, so now I'm done. And so I don't know if there were any questions, or maybe I bored everyone to death.
All right.
Sleep.
So we had a question from Robert Kahn that came through privately to me, who asked who produces yeast catalogs as described.
Oh, so a good one is Scott Labs, and I know during my noon presentation, Chris actually put up a link to it. I could maybe find a link for you, but if you look at Scott Labs Fermentation Handbook, they have a really great handbook. Or you know, if you go to any of the yeast producers' websites, so Lalamond, you can go to their website, and they have a chart that has all the attributes of these yeast, and you can sort of narrow down your selection based on things like ethanol tolerance and temperature tolerance and that sort of thing. Anyway, so I hope that answers your, another good one is Anchor Yeast. So Anchor, Lalamond, and Lefort are the three main companies that produce yeast. So you can look at those catalogs.
It looks like Chris suggested maybe that you might want to talk a little bit about co-inoculation, since we have some time, co-inoculation and versus why we build up the starter as opposed to just dumping yeast in.
Are you talking about building up the starter or adding the malolactic?
Chris, you want to clarify?
Yeah, I think that's what he meant.
Two questions, he says.
What?
He said there are two questions there.
Oh, there's two questions there. So kind of what we. So the co-inoculation is actually a pretty interesting thing that that's starting to become used more and more, and it's co-inoculation with yeast and malolactic bacteria. So if you know that you want to put a wine through malolactic fermentation, so since 99% of red wines you're going to want to do a malolactic fermentation on, it's really important. Sorry. Anyway, I'm sorry, Chris. I thought I heard you ask something. Never mind.
Oh, sorry, I was typing. I should mute.
So in the past, they used to say that you wanted to, before you started malolactic fermentation, you wanted to make sure that all the sugars were removed from your wine, or were taken up from your wine. because malolactic bacteria have the capability of transforming sugar into acetic acid. And so they said, you know, if you have any sugars left, you're going to increase your volatile acidity in your final wine. And now we know that actually when you add the malolactic bacteria at the same, right after you add the yeast, that the yeast preferentially take up the sugar and the malolactic bacteria will preferentially use the malic acid over the sugar. So there actually isn't that, you don't have to worry about that conversion of sugar to acetic acid if you do this co-inoculation. And some of the benefits are that you'll have a quicker malolactic fermentation because you get it started earlier, and you don't have this long period after fermentation is over where you're waiting for malolactic fermentation to finish. It also, some of the off aromas that are produced during malolactic fermentation are absorbed by some of the yeast leaves at the same time that they're being produced. So your wine you end up guarding some of the inherent aromas of the grape, some of these fruity, fresh aromas that you want to keep in your wine. So anyway, so most of the time, you know, now the recommendation is mostly for this co-inoculation, where it's anywhere from 24 to 48 hours after you inoculate with yeast, you add your malolactic bacteria.
Okay, another question came in privately asking how long will yeast live? So before you answer that, I want to post a link to our post-webinar survey and encourage everyone to go to that once we're all finished. So anyway, how long do yeast live?
I assume, are you talking, is it about the dry yeast? Like how, or?
I don't know. He didn't clarify the question. He says yes, the dry yeast.
So the dry yeast. As the dry yeast packages start, after they're opened, they're, you know, kind of the rule of thumb is that they should be used that year. We've used some of the yeast packages in following years, and they seem to ferment fine. They do lose some of their efficiency over time because they get some water absorption. You know, it all depends on how they're stored. They should be stored in the refrigerator, and it should be stored dry, and you might be able to get 2 seasons out of the same yeast package, but for the most part you'd say that they're not as viable the next year.
Okay, it looks like that's all the questions that we had. So Katie, thank you very much for your time tonight. Thanks Chris also for your time. Thanks everyone who attended tonight's webinar. It's really great to have the participation that we do. Just a few final notes. I posted the 2013 webinar schedule for you all to take a look at. And again, please do go take our post-webinar survey that I posted a link to. It really helps us to improve our webinars in the future. So with that, thanks again everyone, and I hope you all